Hello, my children. Welcome to another riveting episode of the Miss Simon's Biology Show. Let me close this music. All right, let's get into it. Today is lesson 2 of Unit 8. It is the last lesson in Unit 8, Cellular Transport. Okay, let me stop with that because it's actually bad. Let's just get into the lesson. So today's lesson objective is that students should be able to describe the process of osmosis in cellular models. Students will be able to differentiate between the different methods of cellular transport. So obviously we have again our plasma membrane, aka the cell membrane, aka the phospholipid bilayer, aka the thing that protects your cell and maintains homeostasis. Inside of the cell, there's a watery fluid, and outside of the cell is floating in watery fluids in your body. So we have to be able to transport certain things. So, for example, salt is used in muscular contractions. We have to transport hormones, all kinds of stuff. All those things are done in this process known as cellular transport. So let's get a little bit deeper into it. So, like I said, a real life analogy is that schools have a gate that surrounds it and basically regulates who comes in and out. Well, cells have a plasma membrane that surrounds the cell and allows good things to come in while stopping bad things from entering. We also learned that diffusion is a naturally occurring process. It occurs very naturally. For example, if you were to put a drop of one chemical, this chemical is a yellow dye, inside of another chemical, then they're going to diffuse, they're going to intermix so they can spread out as much as possible. Same thing with if I were to spray my Lysol, right? It doesn't stay right here where I sprayed it out. It's going to spread around the room. And whatnot. That's a naturally occurring process. Things always want to go from a high concentration out and spread out into a low concentration. Okay, this happens in your cells. This happens in everything that you smell. As always, there is going to be some sort of semi-permeable membrane that the molecules can cross through. And naturally, there's going to be a concentration gradient. One side has to be higher than the other and whatnot. So that brings us to our two types of transport: passive transport and active transport. As their names suggest, passive happens passively. Like if you've ever seen those TikToks where they talk to you about making passive income, that means making money without working necessarily. That's making passive money. So passive transport happens without energy. That's going from high to low. Remember, we know that happens naturally from high to low. There are three types of passive transport that we'll talk about in this class: diffusion, osmosis. Osmosis is literally just diffusion of water. And then facilitated diffusion, which is just diffusion, but it has a little channel helper. Then we have what's known as active transport. Active transport, as the name suggests, is going to require some energy, such as if you were to get active. There's two main types: endo and exocytosis. Endo, bring in; exo, push out. Sorry if I'm talking fast. Hopefully this is a recap. So again, passive transport is from high to low, and active transport is from low up to high. Okay, passive, high to low. Active goes from low to high. So here's what that might look like. So diffusion, again, is from low to high. So as you can notice, other way. That's the other way around. Pause. Pause the video. The cameras. Take two. Diffusion goes from high to low. And then we have active transport that goes from low to high. Okay, so diffusion, high concentration of this type of molecule diffuses through. If it needs a channel by chance, but it's still going from high to low, that's known as facilitated diffusion. If there's any instance in which we are forcing the low concentrations into an area of high concentration, we have to input energy, which comes in the form of adenine triphosphate, aka ATP. Okay. Let's make sure that you kind of understand this topic. Diffusion or osmosis. Look at this first example. So Maddie, the girl who sits at the back of your math class, smells like a walking perfume shop. Would that be diffusion or osmosis at play? Diffusion. It's talking about a smell. Okay, next one. Gargling with warm salt water when your throat hurts causes your swollen throat cells to shrink and hurt you less? Diffusion or osmosis? Osmosis, we're talking about water. After swimming in your friend's house all afternoon, your fingertips get wrinkly. Diffusion or osmosis? Osmosis, we're talking about water, we're talking about swimming, okay? So that's what I mean when I say diffusion or osmosis. If it's talking about water moving, then we are talking about osmosis. If we're talking about anything else moving, it's diffusion. Osmosis, again. Water is always passing freely through the cell membrane. It's quote unquote on the list, right? If the cell membrane is the bouncer outside of a club, then water is always on the list. It's always allowed in and out. And as a result, maintaining water balance is extremely important in maintaining homeostasis of the cell and organism. You probably have heard that our bodies are what, like 78 or something percent water? Well, that lets you know just how important this concept of osmosis is to your cells. So sometimes your cells might have 20% glucose, while the watery environment that it's in may only have 10% glucose. Where does water go? Well, let's find out together. Water is always going to want to diffuse towards the highest concentration of solutes. What is a solute? Well, we could be talking about salt, we could be talking about glucose, we could be talking about potassium. It doesn't matter, okay? The solute is basically the thing that is not water, whatever chemical is we're talking about. Okay. In an effort to reach what is called dynamic equilibrium, which means that yes, we will have half over here on this side of the membrane and half of it over here on the other side of the membrane to reach equilibrium. But dynamic equilibrium just means that they're still moving, they're just moving in equal ways. So at no point does water move and just stop. It does what's known as dynamic equilibrium. So it will be equal on each side. Like if you were to take a snapshot, it'll be equal on each side, but it's dynamic. They're still moving, right? Molecules rarely ever stay still. So, so we're in the situation where water go. Well, if water goes towards the highest solute, then water will probably go either to 10% or to 20%. Which one is higher? The 20%, which means that water is going to flow into the cell because water always goes to wherever there's more solute. So this is the handy dandy thing. Water follows solute. Water is either going to move into a cell or out of a cell, depending on the type of solution that the cell is in. That's where we get this concept of tonicity and the hyper, hypo, iso, all of that fun stuff. So solution concentrations. Hypotonic. A hypo means below. Hypo, below. That means that the solution is low, right? The solution is hypo, it's low, which means if the solution is low, then the cell must be high. Okay, next one. Isotonic. Iso just means same. If you ever done isometric contractions, which is the type of workout, you know you're supposed to keep all of your muscles contracted at the same intensity to try and train them. Isotonic means same. So the solution concentrations and the cell concentrations are the exact same. Last but not least, we have hypertonic, which, if you've ever been hyper on sugar, then you know that hyper means a lot. It means extra. So the hypertonic solution means that there's lots of solutes in the solution and there's less solutes in the actual cell itself. Okay, so notice whenever you use these words, hypotonic, isotonic, hypertonic, you may think that we're talking about the cell. We are not. It is a hypotonic solution, isotonic solution, hypertonic solution. Okay, make sure that you always know that there's solution at the end. So let's look at some examples to try and really clarify this. So hyper, hypo, or iso. So we have 3 different beakers here. We're going to go one by one. Let's start with beaker A. So beaker A tells us there's 10% salt in the cell and there's 10% salt surrounding the cell. Would the solution be hypo hyper or isotonic to the cell? Isotonic. It's the same, 10 and 10. They're the same concentration of salt. Next problem, beaker B. We have 10% salt in the cell and 2% salt surrounding the cell. Would that be hyper, hypo, or iso solution? Hypo. 10 is big, 2 is little, and we're always talking about the solution. So the solution is low, the solution is hypo. Beaker C. What about this one? What do you think it is? Hyper, right? We're looking at the solution. The solution is 30, the cell is 10. 30 is greater than 10, which means 30 is hyper. Hyper is higher than the cell. Water follows solutes. Remember that, please. Water will always move towards whatever side has the higher concentration of solutes. So what we just did was we determined what the solution was. Was it iso, hyper, or hypo? Now we need to determine where is water going to flow. In the cell, out of the cell, or both in and out in dynamic equilibrium. So we have iso. What do we think? Where's water going to follow? It's going to move in and out equally because these are the same, right? They're isotonic. So they're the same. All right, how about beaker B? 2% versus 10%. Where's water going to flow? Water is going to flow towards the higher concentration, which is 10. Water is going to flow in. All right, last but not least, where do you think water is going to go? It's going to come out of the cell towards the 30, so water is going to flow out of the cell. Okay, let's do some more examples of this. Let's just keep practicing. So I just made up an experiment. Notice that I have different units on it. ppm just stands for parts per million. You don't necessarily need to know that, but just in case you encounter it in the future. So we have a hypotonic solution. So the solute concentration is low. And the cell concentration is high, right? 2 is way bigger than 0.005. Water is going to follow solutes. So where does water go? Water is going to flow into the cell. That means there's going to be extra water in the cell, and the cell is going to blow up like a little water balloon, and it might even burst. Okay. Next problem. What type of solution is this? Yep. Solute concentration is the same, 100 and 100. Water follows solutes, and where does water go? It's going to flow in and out of the cell equally in what is known as dynamic equilibrium. The cell is going to behave normally. Last but not least, this is a hypertonic solution, right? 200 is bigger than 2. Solute concentration is high outside of the cell, and the cell concentration is low inside of the cell. Water is going to always follow more solutes. So water is going to flow out of the cell, and that will make the cell shrivel up. Okay? So when water flows out of the cell, obviously it's going to shrivel up like that. When water flows into the cell, it's going to get really big and might even burst. And then if something is isotonic, then the cell is just normal. So some practice problems. So the inside of the cell has 45% NaCl. Just kidding. NaCl, salt. The outside of the cell has 46% NaCl. I can't not say NaCl. I'm sorry. The solution is blank tonic, and water will flow blank the cell. This is normally how your problems are going to look. Now, you might think that you're big and bad, and you might try and do this in your head. I don't recommend that. The reason why I am the super genius that I am is because I know that I know that drawing a picture is always going to help me out. It just is. Like, it doesn't have to be anything fancy. Like, I literally just will scribble things and figure it out. So let's draw a picture for this problem, just so I can show you how to solve it. So boom. First things first, we're going to draw a cell. inside of some sort of solution. Then we're going to put our numbers from the problem. So it says the inside of the cell is 45, it says the outside of the cell is 46. Still with me? Good. For my sake, I always like to make the number that's bigger, I just write it bigger. Even though it's only is like 1% bigger, it's still the bigger number, and I feel like I'm less likely to make a mistake if I just write that number really big. It works. Okay. So now we can tell that water is going to flow, or first, I'm getting ahead of myself. The solution is higher, higher is hyper, hypertonic. Water will flow blank the cell. What's our rule? Our rule is that water follows solutes. So water is going to flow towards this one. All right, now we're going to do some practice problems. You're going to do them 100% on your own. I really want you to try your best. And luckily, I've already drawn pictures for you. So actually, let's do this one. So pure water is going to be 0% solute. It's pure water. So can it have any solutes in it if it's pure water? No, it's 0. Then we have a 5% salt solution. Okay. All right, now really try this one on your own. Here's how to solve it. Next problem, go ahead and do it on your own. Here's how to solve it. Always write your numbers. Okay. That's basically the most simple way that the problems will look, but more often than not, they're going to look like this. I know it looks like it's a lot, but it's honestly very simple information, and you already have been doing awesome, so you keep doing awesome. Let's try this together. So a student is going to place 2 eggs in vinegar to dissolve the eggshells. In order to investigate osmosis, he then placed the equal-sized eggs in the solutions listed in the table. Solutions, initial mass of the egg, and then the final mass of the egg. You can think of mass, if it's easier for you to think of mass as the weight. So obviously a higher mass is going to be a heavier egg and whatnot. What can you conclude about the solutions from the change in mass of the eggs? So basically we need to determine corn syrup. Is it hypo or hyper? Distilled water, is it hypo or hyper compared to the egg, which is our cell? Egg, which is our cell. There you go. Okay, so let's figure out how to do this. So the question is asking us about the change in mass of the egg. So what do we notice about the change in mass? For corn syrup, did the egg gain or lose mass? And for distilled water, did the egg gain or lose mass? Well, corn syrup made the egg lose mass. Distilled water made the egg gain mass. So right now you might already know what the answer is, but you might not. So I'm going to go through the next step. So if you still don't know the answer now, let's go through the next step. What do we do next? Well, next we draw pictures. So I have my egg, which is my cell, and I have my beaker, which is filled with either corn syrup or water. And I'm going to basically just diagram what we just saw. So corn syrup made the egg lose mass, and water made the egg gain mass. Okay, we're talking about osmosis, and with osmosis, we know that water follows solutes. Then, boom, we have it right here. Water follows solute. So if stuff left the egg, that meant that it was following the solute. So it means that the higher concentrations were out here. Which means that corn syrup was hyper, right? It was higher. Distilled water made water flow into the cell, which means it must have had more solutes here. That meant that the water solution had less solutes. Less solutes is hypotonic. If you don't understand that, pause the video, replay the video, because I promise you, you can get it. You probably got it though. I'm sure you got it. Okay, next problem. So the diagram given shows animal and plant cells placed in 3 different types of solutions. What type of solution is represented by diagram C? So we're looking at diagram C. What conditions are we talking about here? Hypotonic. If the cell gained a bunch of water, water flew in, that meant there was more concentrations in here because water follows more solutes. If there's more in here, there must be less out here. If there's less out here, it is hypotonic. Image below represents a cell with a semi-permeable membrane. The process of osmosis would explain the net movement of water into the cell if the initial percentage of... So with this one, something to note that they will try and trick you. So this, I believe this is a STAR question. They will try and trick you and make it difficult because they know that this concept is really easy. Make sure that all of your answer choices are in the same units. So if answer choice A is talking about protein, you can't compare that to answer choice B that is talking about water. You need to make sure that they are the same units. So you just convert them. So for example, A, if protein is 35%, then that means that water must be the other part of the 100%, if that makes sense. Like this. So to avoid confusion, you want to rewrite the answer choices so they're all talking about solute concentration. So before, water was 95% inside. That meant that protein is 5% inside, right? Water was 90% inside the cell. Protein is 10% inside. That makes sense? So everything has to add up to 100. Yeah, y'all get that. That's very simple math. So now you can accurately go through it. Now that they're all talking about protein. So now answer the question. All right, last but not least, remember we're talking about a hypertonic solution. Water is going to go towards more solutes, which means it's going to come out to the solution. Isotonic, they're going in and out, dynamic equilibrium. Hypotonic, water is flowing into the cell might even burst. It's crazy. Cool. All right, last lesson is going to be lesson 9.1, photosynthesis and cellular respiration. Please make sure that you do your exit ticket. Thank you very much. Thank you very much. Thank you.